to put in a disclaimer that this is a very difficult talk to present because there are people in the audience who work in the same line that I do and who know some of these details, but would like to know the technical details of this. And there are people in the audience who are from completely different domains who would like to know what this domain is all about, but they don't want the technical details. So as you know that this is a very difficult talk to give. I will try my very best. I'm going to go bang, 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 meaning that go 100 miles an hour to cover as much of the road as I possibly can without being overly simplistic. So I'm going to start with a couple of stories to sort of begin my talk. And my story number one, this is from the pages of a very renowned journal called the New England Journal of Medicine, one of the oldest journals in medicine. And this story is as follows: that there came a person with a profile such as this, where each dot represents a tumor, and this person has tumor that has spread all over the body. So this this patient came to the doctor, and the doctor treated this person with a specific kind of medication at a specific dose for about 3 weeks or so. And later, 3 weeks later, when the whole body imaging was done, this is what happened to that individual. All of the tumors were actually gone. This journal also described another patient who essentially came with the same kind of profile, and the same medication was provided to that person at the same dose level for the same duration, and 3 weeks later this person remained in exactly the same state as he had appeared at the time when he presented himself to the doctor. So the question that we shall ask is, why is there a smiley here and a sad face here? We'll revisit this story at the end. The second story that I'm going to share with you is a personal encounter, and this is a teacher of mine. I studied at the Indian Statistical Institute. This is a teacher of mine who taught us geology, was a chain smoker, and ended his life in a little sad way. So this is a conversation that I overheard. He had come down with lung cancer. I knew the family. The wife contacted me because I work in this area. I know a number of oncologists in the city, cancer clinicians in the city. So I put her in touch with one of the cancer clinicians, and the clinician and the wife of the patient, my teacher's wife, are walking, and I'm sort of shadowing them at the back. And this is the conversation that I'm overhearing. The doctor says, well, the patient has non-small cell lung carcinoma. Don't even worry about what non-small cell is. The patient has lung cancer. I need to put him on chemotherapy. Immediately, the tentacles go up in the wife's mind, and essentially the wife says that can the chemotherapy be done at home? They live about 50 kilometers away from where the hospital is, and she's thinking that if the chemotherapy starts, she has to go up and down the 50 miles every day, traverse 100 miles in order to be on the bedside of the husband, and that's not easy in the city of Calcutta. So she asks this naive question, simplistic question: can the chemotherapy be done at home? The doctor's immediate reaction is no, but then he takes a pause and says, wait a minute, let's check out his EGFR mutation status. I'll come back to what EGFR is. It's a gene in the genome of this patient, and he wants to check out the mutation status, meaning if there are alterations in this gene. And then the doctor goes on to explain that if it turns out that he has EGFR mutations, then you can take him home and administer chemotherapy orally. So we'll come back to find out whether the patient did have EGFR mutations and what happened to this individual and why this doctor made the statement that he did. Cancer, as many of us know now, is a disease of the genome, meaning that there are alterations in the genetic material in the cells of our body. When we talk about a disease of the genome, we believe that it's going to aggregate in families and it's going to be passed on from one generation to another. That does not happen in cancer, but one of the reasons why we believe that cancer is a disease of the genome is that cancer cells are very disorganized. Usually you would have 23 pairs of chromosomes aligned very well as pairs, but here is what you see. There are many chromosomes that have multiple copies. Bits and pieces of chromosomes have traveled around from its normal place to an abnormal place, etc. The hallmark of cancer is unregulated cell growth. So this is what happens in cancer cells. They are very disorganized. They have an unregulated cell growth. Usually in the normal body, cell growth is very regulated. From one cell we get 2 cells. From 2 cells we get 4 cells. This is not what happens in cancer. It's completely the complete dysregulation of the cellular state. Like I said, that when we talk about a disease of the genome, we think that it is going to be familial, it's going to be passed on from one generation to another. In cancer, that does not happen. Only in about 10% of cancers is it familial, and these cancers have been very well described over the last 50 or 70 years that we've done genetics on cancers, and these familial cancers have actually been described very well. For a long time, we had to take a pause, primarily because we did not have the arsenal, we did not have the technology to even think about what may be the genetic basis of non-familial cancers. About 15 years ago, there arose these interesting DNA sequencers called next generation sequencers. I will tell you a little bit about these next generation sequencers that enabled us to actually dig deeper and find out the non-familial nature of cancer. So essentially what I'm going to do is to tell you about our foray into the non-familial nature of cancers and the understanding that we've had. I'm not going to talk about familial cancers. I'm going to introduce two terms here. One's called, well, you know, we all know what genes are. The entire genome contains about, well, I'll introduce that, contains about 3 billion nucleotides, and only about 1% of the genome produces proteins, and those proteins are important for our regular activities, regular functioning of the body. We have learned a lot about familial cancers and the genes that are altered in familial cancers, but we hardly knew anything about non-familial cancers, and that's what I'm going to primarily talk to you about. One of the persons, like I said, that we couldn't have done this without the sequencing, and sequencing came under big fire about 20-25 years ago. People didn't even believe that we needed to sequence the genome, primarily because of the costs that were involved. Scientists in other domains felt that the biologists and these technology pushers are going to actually take away all the money from all of science, and therefore it wasn't necessary. So there was a huge outcry because of the costs involved. But this is a Nobel laureate, Renato Dubecco, who said that who wrote a paper in Science called A Turning Point in Cancer Research, Sequencing the Human Genome. And one of the things that he said, and I read it out for you: if we wish to learn more about cancer, we must concentrate on the cellular genome. We have two options: either to try to discover the genes important in malignancy by a piecemeal approach, or to sequence the whole genome. It will be far more useful to begin by sequencing the cellular genome, because we will discover new things, we will be able to find cancer genes faster. And this is the reason why sequencing the human genome has led to many interesting scientific discoveries, some of which I will describe to you. If you look at the human genome, like I said, we are diploid. We have 23 pairs of chromosomes, 22 pairs of autosomal chromosomes, 1 pair of gender-determining chromosomes, and the number of alphabets, this is a linear string of alphabets. The number of alphabets is 3.2 billion if you look at the haploid set, and if you look at the diploid set, it's 6.4 billion nucleotides. We also sometimes do not study the entire genome. I'm also going to use the word whole genome sequencing and exome sequencing. When we talk about the exome, this is about 180,000 exons of genes that participate in the production of proteins that constitute about 1% of the total genes. In other words, if you're going to sequence the exome, we have to sequence 3 million nucleotides as opposed to 3 billion nucleotides. Many of us believe, and that belief is actually sort of falling by the wayside as we are discovering newer things in the genome. Mutations in the exome are thought to harbor about 85% of the mutations that have a large effect on disease. It used to be 85%, but it's actually receding because there are other alterations in other parts of the genome. Non-exomic fraction of the genome are also being seen to have a very large effect on various kinds of diseases, including cancer. One of the things that we do, I said that this technology arose about 15 years ago. Essentially, what we do is for people who are not biologists, what we used to do is to look at a particular region of the genome and make multiple copies of it artificially in a water bath. That artificial amplification of that specific segment of the genome led to loss of different kinds of alterations that were harbored by different cells in the body in that particular fragment, and therefore that technology would not discover various kinds of alterations that took place in different body cells. So we had to wait for another kind of technology to arise where we could actually look at that particular fragment in a large number of cells in the body without losing any of the alterations that took place in a specific cell or in a group of cells. So that particular technology is called next generation sequencing, and we use that in order to be able to identify all of the cellular components, all of the cellular alterations in the genes that we are looking at, or even across the entire genome and associated with cancer. These sequencing platforms have now been standardized. We can actually either get the whole exome sequence or get the whole genome sequence and at depth, so that we don't lose any of the variants that are available, that are present in some of the cells in the human body. This is easy to generate now. It's become easy to generate these kinds of data. Just to give you an idea, the first human sequencing using to get the sequence took about 13 years. And now I work in an institution in Kalyani called the National Institute of Biomedical Genomics, and even in that remote place in India, we can actually sequence about 6 genomes a week. Compared to the first genome that took 13 years. The important part is not to be able to generate the data. The important part is to be able to do this computation, or the more difficult part is to do the computation and the statistical analysis of the data. I do not have the opportunity right now to either explain this this portion of the slide in any detail, or this portion of the slide in any detail. You will only bear with me that when we generate data on one patient, we generate about 200 terabytes of data. So that's the kind of data that we generate from each patient. We usually analyze DNA in the tumor cells, and we analyze DNA either in the adjacent normal cell or from the blood. So from each patient, we generate 100 terabytes of data from each of these two components, and so the total amount of data that we generate for each individual patient is about 200 terabytes, and we look at a large number of patients in order to draw statistical inferences, and that's what I'm going to describe to you. So we look at the evolution of the cancer genome, and please remember that we are not looking at the germline, we are not looking at the heritable component of the genome, we are looking at somatic component, which is not passed on from parents to their children. So this happens in the individual. Whatever changes are happening in the individual will die with the individual and will not be passed on to the next generation. So these, imagine if you will, that these are cells in the body. So this is like the cells under my skin. So this is the epithelial layer of the skin, and these cells look exactly alike as we are born. And these cells, as we undergo our life course, these cells, some of these cells, one or more of these cells will accumulate alterations. So imagine, if you will, that these 3 cells have gotten some kind of alteration, and the difference in color actually tells us that these are different kinds of alterations in the genome of the individual in those specific cells. Some of these will give the cell a slight growth advantage. So there are other kinds of mutations that appear on the background of these cells. Some of these mutations will give the cell a slight growth advantage, meaning, like I said, that usually it's 1 to 2 to 4, etc. It's like a binary fission that happens in the normal cell division. Here, with this particular mutation, after it arose, it provided this particular cell a peculiar advantage, in which it's not going to grow 1 to 2 to 4. It actually made a clone of 4 cells now. So this is just photocopying process. It's not the usual way the cell division takes place. So this particular mutation gave rise to that ability for the cell to have a peculiar growth advantage, and that growth advantage led to a small clone of cells, and this clone of cells is called a tumor. But this is most of these tumors arise in the body. They are benign tumors. We don't care about them. But sometimes we do care when we have cancer. So here is another mutation that took place that led to another clonal growth. And again, we are completely oblivious to such things happening in our body. And believe me, that they are happening all the time in the body, and we really don't care about them. In the back, so such mutations will keep accumulating. So here is another mutation that arose on the background of these 2 mutations, the black and the yellow. And this particular mutation may or may not give any growth advantage to the cell, but this particular mutation did. And what it did is that it gave the cell a huge growth advantage, in the sense that it breached the epithelial layer and became a malignant tumor. The remaining, the previous mutations that took place gave the cell a slight growth advantage, but did not actually acquire the properties of a huge growth advantage such that it becomes a malignant tumor. So we are interested in such mutations, and at the background of these, more and more mutations will accumulate, more and more genetic alterations will accumulate. So the question that we ask is, what are those genes and genomic alterations that drive? And that red mutation, that red alteration that I showed you, is actually the one that drove this particular cell to becoming a malignant tumor and gave the cell a huge growth advantage. And those are the ones that we are actually calling as drivers, and those are the ones that we want to identify. You might imagine that it may be easy to identify these drivers, but if you think about it a little bit, it's not easy to identify the drivers, because this is happening at the background of all of this noise where there are these alterations, and in the background of all of these noise, we are supposed to identify this signal, which is really the driver that leads to cancer. The reason why we want to identify these as opposed to these is because if we can identify those, and if we can pinpoint which particular gene that alteration is happening, then maybe we can think of certain kinds of therapies, and also may be able to prevent the cancer. Okay, so I'm going to describe to you our own work on oral cancer. Oral cancer is the 8th most common cancer in the world. About 250,000 cases, new cases arise annually. 2/3 of them arise in developing countries. About 130,000 deaths occur annually. It accounts for about 1/3 of all tobacco-related cancers in India. So oral cancer among the males is the most prominent form of cancer in India. And of course, we know what the hazardous combination is. It's essentially the tobacco, but the snakehead lion chews up the epithelial layer and exposes the cells directly to the carcinogens that are available, that are present in tobacco. Areca nut also has certain kinds of carcinogens. So this combination is the hazardous combination that leads to oral cancer, or precipitates oral cancer. It also leads to certain kinds of changes in the genome that eventually changes in the somatic cells in the genome that eventually leads to a driver mutation that produces a huge clone of cells and becomes a malignant tumor. We work with patients. Our patients we are right now about 500, but I'm going to present data to you that are on 300 patients. Most were males. About 72% of the patients were middle-aged. 86% of the patients presented themselves very late to the clinician. This is called stage 4. The reason why they present themselves very late to the clinician is that usually they'll get a bruise, they'll get a small tumor in the inside of the mouth, but they love their tobacco so much that they will not go to the doctor. So they will keep chewing tobacco until they can bear it no more. And then when they go to the doctor, it's kind of late. It's already stage 4. 86% of our patients presented themselves very late. Most of them were exposed to tobacco, 80% of them for greater than 10 years. So the few patients who said that they were not exposed to tobacco, actually we figured out tobacco leaves a telltale mark on the genome. We actually figured out that they lied to us because the same telltale mark was also present in their genome. About a quarter of them harbored the human papilloma virus. So what we did was a combination, and I don't have the time right now to separate the two, so I've mixed the two. We did both exome and whole genome resequencing, and we've done this on 300 individuals, and it's always paired blood and tumor DNA. So we take take a biopsy from the tumor, isolate the DNA from those cells in the tumor, and take the blood of that individual. The blood of the individual contains germline mutations that have been inherited from the parents. The tumor DNA contains, of course, those germline mutations that have been inherited, plus the life course mutations that happen during the life course of this individual in the cells that will not be passed on to their offspring, but they are present in the body cells of that particular individual. So if the tumor cells is a union of both the somatic mutations and the germline mutations. The blood samples contains the somatic mutations. So if we subtract out the mutations that are present in the blood from the union of this germline and the somatic mutations, we get a repertoire of only a list of catalog of only somatic mutations, and that's what we want to deal with. We want to deal with the somatic mutations. We want to statistically analyze the somatic mutations in order to identify the drivers, and that's the reason why we do for each patient a paired blood and tumor DNA analysis. If you look at, so I'm presenting you data on about 300 patients. So these are the 300 patients that we're looking at. The first point to note is that the number of single nucleotide variants. So single nucleotide variants are change of one alphabet in the normal genome to another alphabet in the patient genome. So those are called single nucleotide changes, single alphabet changes, and they're the most prominent kind of changes in the human genome that take place. These mutations, alterations that I'm talking about are mostly single alphabet changes. There are other kinds of changes. If I have the time, I'll come to those later. But these are the most important kinds of important. I don't know, most prevalent kind of changes that take place in the human genome when we deal with diseases such as cancer. So if you look at patients, each vertical bar represents the total number of mutations in that patients. We have categorized the mutations by certain subclasses, but don't worry about it. The main point that I want to tell you is that some of these patients have small number of mutations and many of these patients have a very large number of mutations. The total number of single nucleotide variants is highly variable across patients of oral cancer. If you look at these variants, these variants can happen in can take place in two different kinds of genes. So let me introduce to you the two different kinds of genes that are important in the context of cancer. One's called a tumor suppressor genes. These genes usually express certain proteins that prevent the formation of cancer, that prevent the formation of a tumor. These are called tumor suppressors. They are always awake and they are producing these proteins to prevent cancers from happening in the body. But when, because of an alteration, a single nucleotide alteration or any other kind of alteration, this gene is made non-functional, it essentially goes to sleep and it does not produce that beneficial protein that will protect you from these kinds of cancers, from cancers to appear in the body. And therefore, tumor suppressors are usually, the point to note is that tumor suppressors are usually awake. Because of certain kinds of DNA changes, they will go to sleep and they will predispose you to certain kinds of cancers. The other is, so this is a mutated tumor suppressor gene where it will stop producing the protein and it will provide some kind of a strange kinds of cellular division. The other kind of mutation, the other kind of genes are called oncogenes, and these genes are usually asleep. They don't produce any proteins, but certain kinds of DNA alterations will wake them up and then they will start producing some rogue proteins, and these rogue proteins will provide cells with a growth advantage and they become clonal and they become a malignant tumor. So these are the two classes of genes that are known to participate in the process of cancer. One is always alive, going to sleep is detrimental. The other class of genes is always asleep, waking them up is detrimental. So we'll see what happens, and I'll make a few comments on the nature of the genes. So what we did, so in order for cancer to appear, tumor suppressor genes are turned off or oncogenes are turned on. When we looked at our data, when you look at data on these 300 patients and we analyze the data statistically, and remember we're only looking at the somatic component because none of these patients had any familiarity of the cancer that we were looking at. What we noted was that, well, these are the names for those of you who are in the business. You will some of these names will be very familiar to you. The one name that may not be familiar in the context of cancer is FAT1, but FAT1 is now becoming very, very important in not only in oral cancer. We discovered that in oral cancer, but it's also now becoming important in other forms of cancer. The important point to note is that after we identified the driver genes, so mutations in these genes drives a cell to becoming a malignant tumor. So these are called driver genes. They are significantly mutated in oral cancer patients. These genes, if you look at the properties of these genes usually express beneficial proteins, and when the mutation happens, they go to sleep, and then they stop producing this beneficial protein, and it makes you vulnerable to predisposed to these kinds of cancers, including oral cancer. And these are tumor suppressors. So essentially, the take-home message is that the genes that drive oral cancer are tumor suppressors, and that's really bad news. The reason being that if a gene is awake, and mutation in any part of the gene or most places of the gene will actually make them go to sleep, will actually make them non-functional. To revive that gene to start producing that protein means that you have to activate that particular alphabet that was changed from the normal to the abnormal, and that kind of change is very difficult. To position ourselves, to anchor ourselves on one particular alphabet and make a change to revert back to the normal is very, very difficult. So when we encounter new tumor suppressors driving a particular cancer type, it's bad news because we can't really control the cancer by reactivating the gene, so to say. So cancer, this is one major finding that we had. And then what we did were, find mapping all of these genes and trying to figure out what kind of patients, what kind of properties do patients have if they have mutations in one gene and not the other, etc. So we were able to figure out, this is the take-home message, we were able to figure out that there are, based on the mutational profile, we can group patients into certain subgroups. What good does it do to group them in certain subgroups? We were able to relate with post-treatment survival. So if you have mutations in certain kinds of genes and if you're treated in the usual way, which is surgery followed by a certain amount of chemotherapy, you survive longer. So post-treatment, post-treatment survival depends on which branch of the tree, and the length of the tree, the length of this line or this vertical line is essentially proportional to the length of time that this person will suffer or will survive without any intervention post treatment, post cancer treatment. So as you can see that these bars are not all of equal length. So having mutations in certain genes are more helpful in the sense that after you go through the same treatment, you will be able to survive longer. So that's the prediction as to how long a patient might survive will depend on where we find the gene mutation. This is one of those genes called MLL4 that gives a huge survival advantage. We published this some years ago. I now look at one particular gene called BRAF. BRAF is a major cancer gene that's found in 70% of malignant melanoma. We didn't find it in oral cancer. 70% of malignant melanoma, 30% of thyroid cancer. Melanoma is skin cancer. 30% of thyroid cancer, 10% of colorectal cancer. And the BRAF protein is involved in sending signals from outside of the cell to inside of the cell. And this signal actually directs or modulates cellular growth. And as I said, that in cancer, cellular growth is hugely disturbed, as a result of which clonal propagation happens, as a result of which tumors form. So these BRAF protein is a signaling protein, and this particular protein is very important. It's produced by this gene called BRAF, and if there are mutations in BRAF, it damages this signaling protein, and the entire signaling cascade goes haywire. There's a common mutation in this particular gene called V600E, which is essentially one amino acid, a single nucleotide change that changes an amino acid to another amino acid. It is an oncogene, and there are drugs now available. So there are drugs now available called bemrafenib, which inhibits this gene if this particular mutation is present, has taken place. This is an oncogene which is usually asleep, and as a result of the mutation, the gene gets activated and produces rogue proteins. So what you can do is, because this is a common mutation, if you can get an inhibitor that will attach to that common mutation or to the amino acid in the common mutation in a three-dimensional form, you can actually inhibit the rogue property of that particular protein. You can actually stop that protein from even taking any action in the body. So that's the advantage of having mutations in oncogenes, because they become, at least they're amenable to action. You can think about drugs with respect to that. So this particular patient, so there are two patients that came to the doctor with the same imaging profile. One of them in 3 weeks' time with a particular drug came to this state, and the other individual came to this state or remained in this same state with that drug for the same duration. And the reason is, you can imagine that this individual actually had a BRAF mutation and was provided with bemrafenib, and because the individual had BRAF mutation, and because vemurafenib is an inhibitor of BRAF, the patient responded very, very well. This unfortunate patient had cancer, had a tumor spread all over the body, not because of a mutation in BRAF, but because of a mutation in some other gene, as a result of which the individual did not respond to vemurafenib and remained in the same state in spite of being treated. So this individual had no. So that's the end of that story. That's how we explain why this individual came to this state and why this individual remained in the same state at which he presented to the doctor. And this is the reason why we need to understand what the drivers are. In this particular case, we know that BRAF is a driver. It's an oncogene. It's actionable. Action was taken. A drug was found. And now if a patient comes in with lung cancer where BRAF mutations are very often mutated, the doctor would actually, BRAF mutations are, of course, mutated in multiple kinds of cancer types, melanoma being the most prominent. So what we did was, okay, so unfortunately for oral cancer, we did not have the privilege of having oncogenes as the drivers. Tumor suppressors were drivers. So we said, okay, so we can't take any action on tumor suppressors, but there are a large number of genes that are mutated, which are not necessarily drivers, but also are mutated in a way. Can we act on them? That was the question that we asked. And if you look at these various genes and the proportion of patients who have mutations in these various kinds of genes, the top of the list is, of course, P53. P53 in oral cancer acts as a tumor suppressor and is not druggable. We can't take any action against P53 in the context of oral cancer. But what we then did was to map all of these mutations on pathways, and once we mapped those mutations on pathways, one of the biological pathways that popped up is called the arachidonic acid metabolism pathway. The arachidonic acid metabolism pathway is very interesting because it regulates inflammation and it also regulates progression. More importantly, we know certain components in this pathway. This is the arachidonic metabolism pathway, and there are components in this pathway that are actionable, where we can take some action. So this particular pathway has components that are called COX and LOX, and the COX and LOX are involved in various kinds of activities that are associated with biological activities that are associated with cancers, and many of these are inhibitors to many of these are actually available already in the market. So aspirin, for example, is a COX inhibitor, and there are COX inhibitors and LOX inhibitors that you can find, and so you can take action against some of these components of the pathway and stop that pathway from acting and stop that pathway from working in negative ways in order to produce tumor. So one of the ways that we thought about is that even though we can't take action against the tumor suppressors, these tumor suppressors, in conjunction with the other genes that also show mutations, they act on a specific biological pathway. So instead of acting on that particular gene, can we act on the pathway? And our answer was yes, we can act on that pathway. And so now it falls in the domain of clinical trials, because we don't really have to find new drugs. They're already marketed drugs, so we need to repurpose, but repurposing is also clinical trial. We are not geared up towards clinical trial because there are lots of regulatory kinds of things that happen, but we are in negotiation with some drug companies to see if we can initiate this particular trial. Interestingly enough, individuals who have mutations in that particular pathway have a great survival advantage compared to individuals who do not have mutations. These mutations are like natural inhibitors, so that's what happens. We published this. So let me. I don't know. I think I have 2 more minutes, so I'll just wrap up this conversation that I overheard between the doctor and the patient relative. If you recall, the patient relative says that doctor says that the patient has lung cancer, need to put him on chemotherapy. Can it be done at home? The doctor initially says no, then says, wait, let me check out his EGFR mutation status, and if he has EGFR mutations, you can take him home. So EGFR is a gene that produces a protein. It's a family of receptors. To cut a long story short, it is involved in activation of anti-apoptotic signaling pathways. Apoptosis is a cell is born and it lives for a while and then dies a natural death. Apoptosis is the process of natural death. Anti-apoptotic means that this cell will forget to die, and there are signaling. Even BRAF produces these kinds of signals. So there are various kinds of signaling phenomena that happens within the human body that lets a cell forget to die, and that's an adverse event. If that happens, then EGFR is one of those signaling molecules, and if there are mutations in EGFR, the cell will forget to die and a tumor will form. And there are specific domains that are responsible for activating anti-apoptotic pathways. You do not want anti-apoptotic pathways to be activated. You want apoptotic pathways to be activated so that the cell is born and it dies a natural death. But in this particular case, if mutations are there in a specific domain of this particular protein, it will activate anti-apoptotic pathways and have the cell not die a natural death, and therefore tumors will appear. There are multiple trials that happen, and it turns out that you can actually take action against mutated EGFR. Afatinib is a selective that's orally bioavailable family of these kinds of receptors, ERBB or B kind of receptors, and it blocks that particular protein. This was tried, this went through clinical trials, and if you look at cisplatin, which is the usual chemotherapeutic drug, platinum-based usual chemotherapeutic drug, the efficacy is 23%, the response rate, the efficacy is 23%. But if the same drug that acts against a specific pathway, which is the ERBB pathway, in that particular case, the oral is, first of all, it's orally available. And the response rate is much higher, 56%. Also, the disease progression-free survival is higher with the oral drug compared to the platinum-based drug. So the reason why this individual, the doctor said, let's check out his EGFR mutation status, and then you can provide the orally bioavailable drug or oral chemotherapy, is because of this particular trial where a new drug has been found that's orally bioavailable and the response rate is very high. Turned out that my teacher, who was sequenced, did not have EGFR mutations, and he passed away shortly after he was admitted to the hospital. So essentially what we really are striving to do by identifying these drivers, etc., is to unleash the power of genomics to effect precision medicine and turn cancer into a chronic disease. So let me explain this last phrase to you. When we learned about hypertension, it was deadly, because hypertension would remain uncontrolled, and uncontrolled hypertension led to various kinds of things, including death. But now we have antihypertensives, and many of us in this hall, including myself, must have taken an antihypertensive pill in the morning, and we can go about doing our daily chores, and we do that every day. Some of us may be doing it twice a day, or taking the drug twice a day. So we live with hypertension. Hypertension has become chronic in us. So what we really want to do is to identify these driver genes, such as EGFR, such as BRAF, where if we have a orally bioavailable therapeutic, a drug that will block, that kind of medication is being precise. Not everybody will have BRAF mutations when you have melanoma, or EGFR mutations when you have lung cancer. But if you do, then, and if there is a drug available, then that drug is precise to your genetic condition. And what we really want to do through all of these genomics is to unleash the power of genomics such that medicine becomes more precise and we can turn cancer into a chronic disease. We don't have to really stay on the bed of a hospital to be treated by a doctor. Thank you very much. That's the hope.